Yes, good afternoon to everybody, and thanks for your presence. Today, for the CIMA seminars series, our invited speaker is Dr. Madeline Dukes. She is located in the East Coast, then for her now is a little bit early. And as an introduction, Madeline Dukes has been at the forefront of in situ liquid phase TEM, and since her graduate works, where she earned her PhD in chemistry from Vanderbilt University in Nashville, Tennessee. Her dissertation, Scanning Transmission Electron Microscopy of Target Proteins in Whole Eukaryotic Cell, supervised by one of the founders of liquid phase TEM, Dr. Niels de Jong. She utilized the first prototype design of the prototype of in situ liquid TEM system, called Poseidon. After completing her degree, she joined Protochip in 2011 as an application scientist specializing in situ liquid phase TEM. Her area of expertise is experiment and workflow development, and she collaborates regularly with in situ liquid cell users around the world to develop and test new liquid TEM technique and instrumentation. Okay, now it's time for me to give the floor to Madeline for the talk. Please.
Thank you so much, and I really appreciate the invitation to come and talk about in situ liquid transmission electron microscopy, and tell you a little bit about the technique, where it's been, where it's going. I'll show some examples of different applications and results from across a wide variety of fields, and then at the end, I'm going to talk a little bit about some of the new developments that we're working on in terms of new capabilities for in situ liquid TEM experiments. So, so of course, I'm sure many of you are very, very familiar with the TEM, the transmission electrode microscope, in which a beam of electrons is used to form the image, and it really allows us to get the structural and elemental analysis of a material from all the way from the micro to the sub nanoscale level. Of course, in order to to do these types of experiments, the sample has to be subjected to a high vacuum. And so, these are used again across a wide variety of different disciplines. But in order to really study processes, if we're using conventional TEM, we are limited to looking at snapshots in time of a pre-process, post-process, and then we have to use other techniques to figure out what's happening between, before and after. And so what in situ is really allowing us to do is in situ TEM really refers to techniques that allow us to take a specimen and be able to image it in the TEM with a more natural environment. So not necessarily just the high vacuum, liquid, gas, apply stimuli such as different temperatures, electrical analysis. And what we want from that is to really be able to watch dynamically what's happening in the microscope. And so I always like to use this example of horses before cameras and photography were developed, is that if you look at paintings, pre-photography, the horses all have their legs stretched out when they're running with all 4 feet off the ground. But with the development of video and photography, they were able to watch that process and slow it down and see what was happening. And what you find out is that the horse actually, when it's running, it always has 1 foot on the ground for trotting and galloping and things like that. So that's what in situ, the goal of in situ, and people started to really play with it and to delve into it, was to see what's happening but retain that resolution in the microscope. And so there's so ultimately, what are we doing? We're trying to correlate structural and dynamic structural changes with a behavior due to a stimuli. And so that can be, changes in, grain boundaries, crystallinity, composition, how things are interacting, how they're degrading, how they're growing. And so there's a very wide variety of different types of in situ work that people are doing. There are commercial solutions, there are many wonderful home-built solutions, and it really, a lot of correlative work. It spans, from TEM to STEM to SEM, light microscopy. So there's a lot of different types of in situ microscopy. Certain in TEM, we see a lot of in situ work focusing on particularly materials design, how the structure is affecting function or stability, energy production and storage, what do battery and fuel cell life cycles look like? And for, things like in gas environments, we see a lot of catalysis research. And so how do we do this? And so ideally you're creating a laboratory in the TEM. And so the challenges with that really are things like your sample size. The sample, by virtue of being a TEM sample, is very, very small. So it is not completely analogous to real world, operating material. You have to control the environment without damaging the microscope. So that means if you're working in liquid or gas, you have to find a way to be able to image that environment without that, disrupting the column in any way. Ideally, you want to be able to deliver stimuli, which can be things like temperature, electrical. You want to be able to measure what's changing, know what temperature the sample's at a different, any respective time in the experiment. And you have to think about beam effects, because when we start to add things like liquid or gas, the beam is also interacting with those, and they can interact with our sample. And so in general, for in situ work that is outside, that requires a non-vacuum environment, you're going to utilize a closed cell type of setup, in which, or in some cases, a differential pumping setup. So at this point, I'm going to kind of transition. We're just going to talk just about liquid TEM. So that's when we take a sample that's in liquid, and we put that into one of these dedicated sample holders, and we're going to, do our experiment in the microscope. And so, normally samples that are hydrated, we have to do things to them in order to look at them in the TEM. So that can be things like drying them. That's everything from, biological soft material samples to even, nanoparticles that are maybe in a beaker. We take those out, we put them on a grid, we let them dry, we image them. We embed samples, things like resin. We freeze samples. That's, for cryo and biological materials. That's kind of the gold standard. And so what we want to do in liquid is look at the dynamics. We're going to kind of put a tiny, almost nano aquarium into the microscope. And so, this is not necessarily something new that people have been looking at. So the idea of encapsulating a sample to keep the liquid confined between two transparent membranes really, initiated, almost with the, the development of the TEM itself. So as early as 1935, Laszlo Martin was trying to look at like biological bacterial specimens. In this case, they were sort of sandwiched between two very thick, half micron thick aluminum foils. A lot of early work was done using very, very high voltage microscopes. And then in 1978, looking at a film of colloidin on a colloidin and silicon oxide film on a copper grid, they were looking at the hydration and the development of Portland cement. And so that's almost more analogous, to some of our more modern systems. So they had, the two membranes, they had their cements, their hydrated cement solution. They were able to inject liquid, and they took these images, looking at how those that cement forms. And of course, with things like this, I mean, they're working at, very thick layers. The resolution is not particularly high, but the intent to do this sort of work was always there. However, it really wasn't until the development of semiconductor thin films that we were able to take this to a more practical level, to start to really, take it from being a novelty to something that we could learn real science from. We could see real structural information. We can get much better resolution. And so MEMS-based films or micro electromechanical systems sample supports use traditional semiconductor fabrication techniques. They've fabricated on a wafer, and they've really, revolutionized what we can do with in situ and operando TEM studies, particularly for liquid TEM and gas TEM studies. So these membranes are ultra thin, so they're no more, half micron aluminum foils. They are typically in the 50 nanometer range, generally silicon nitride, but there are other types of films. You can pattern different types of features on them. So you can pattern heating coils, you can pattern electrodes, you can make, all type, put different electrode materials down. And you can do this on a high throughput. So you're able to make, many identical setups to do multiple experiments. And so, of course, these are very small. This is just a conventional setup. We'll use 2 chips. Each chip will have a membrane in it. That sandwich holds the sample in the holder. And so this is just an example of one of the protochip cells. So this would be an electrochemistry cell. We have a small chip, put a droplet of sample or inject a droplet of sample, and then there's a large chip on top of that that has, in this case, electrodes for doing electrochemical experiments. And I'll show you a little bit more about the actual holder itself in the next couple of slides. So this is some of the very early work from my advisor, Dr. Niels de Jong. Initially, he approached it with the idea that this was going to be really, used very widely, ideally in biology. And so this is one of the first holders that protochips made. It's a very simple design. It has 3 O-rings, and I'll show you on the next slide kind of the cross-section of that. The chips sort of sit in a little pocket. They're the same size. You can flow liquid in from the back of the holder here. It flows through the shaft, through that chip stack, which is hermetically sealed with a series of 3 O-rings, and back out. And in this case, they're looking at yeast cells that have some fluorescent indicators. So they would take this holder and put it on a light microscope, look at and see, where the yeast were in the cell, if they had their fluorescence. And so this is the light microscopy and fluorescence microscopy, and overlaid here with the STEM image. So that's some very early work using the liquid cell holder to look at a biological specimen. In general, what we've found over the past decade and a half is because of the extreme beam interactions, that where a liquid cell is really gone is it's not so much focusing on biological materials, but really energy materials, nanomaterial growth, synthesis, degradation, things like that. And so it's really moved more heavily into the material science versus the life science. But there are still some really interesting studies that are focusing on biological and soft materials. And so over the past decade, there's been a lot of evolution of these holders. There's several commercial companies, including protochips, that make really excellent systems. And then there's many labs that will build their own systems. So this is the early design. Very simple. It has 2 chips the same size. It has small O-ring, large O-ring, and another small O-ring. And so the disadvantage with this really is that you have this very small hole here. So one of the big advantages, of course, with TEM is the ability to do chemical analysis. So one of the things that was sort of recognized within the first few years is that this, in order to do things like EDS, was going to need somewhat of a redesign. They wanted to also be able to do more types of experiments. So that meant changing the geometry in order to get electrodes and things into the cell so they could watch electrochemistry. So this is some early work again with, in this case, whole cells, just looking at fluorescence, and then focusing on a small part of that and looking at it in the stem to see the dispersion of nanoparticles of these CAD selenite quantum dots. About in 2011, 2012, the designs really kind of changed, and you see this across systems that went to a large chip, small chip design. So one of the reasons for that was, if you have a larger chip, you can put more things on it. And so this allows you to do things like electrochemistry. In this case, this is looking at a charge discharge of a lithium battery. Also, because with the larger chip, you can eliminate the top O-ring, so you can have a thinner holder, which allows you to take advantage of smaller gap pull piece TEMs. And then finally, working with Nestor Zaluzak and at Argonne and Manchester, the holder, at least with protochips, was redesigned in about 2013 to remove most of the penumbra of the lid with this butterfly design and allow us to do real EDS. So we can do chemical analysis, we can do electrochemistry, we can do some correlative light analysis. And so really, able to do a lot of things with these types of systems. So very briefly, this is what a modern liquid cell system looks like. Again, other companies are very similar. They have the same concepts of some type of sealing O-rings. They all use multiple a chip stack essentially that you can mix and match that has a very thin membrane. In this case, we use 50 nanometer silicon nitride. Integrated into the holder in this case are electrodes. Now the electrodes here connect to the electrical connection at the back of the holder. And by changing the chips that you're using, you can change the function of the experiment. So if you're just doing, say, a liquid only type experiment, you can use chips that just have a spacer so that you can set the thickness of the liquid and flow liquid in. You can mix liquid. You can just do a static experiment. If you want to do electrochemistry, you can select a chip, or if you're really good at in the fab, you can make a chip that has different electrode materials, geometries, and perform electrochemistry in the microscope. And then in this case, if you want to look at higher temperatures, this is a heating chip. So it has an embedded tungsten coil that will resistively heat that whole chip like a hot plate and allow you to look at reactions that are occurring up to 100 degrees Celsius. And so that's a little bit about where the technology has developed over the last 15 years or so. And so at this point, I'm going to talk a little bit about the workflow and the challenges, and then we'll go into what people are using this technique for. So in terms of studies, what I see the most in the literature really can kind of be divided into a couple of buckets. There's a lot of synthesis and growth studies looking at changes in kinetics and morphology, size distribution, and how things are growing and changing under certain conditions. There are a lot of stability studies. Now, this can be both stability of particles as respect to temperature, respect to electrical cycling. And then, of course, there's true operando studies where they're looking at, correlating, electrochemical or catalytic behavior, to specific structural changes that occur. And so with in situ, the in situ workflow is a lot of iterative testing. So you develop your in situ experiment, you analyze everything before you put it into the holder, you perform an in situ experiment, you adjust things, and you do ex situ and correlative tests, then you repeat. And so it's very rarely sort of a one and done type of technique. So this is a nice example here of looking at some a commercial fuel cell catalyst where they've done very nice high resolution characterization of the material before it goes into the in situ cell. Then they prepare their in situ cell. They're doing their electrochemical cycling. They're watching in this case, the coarsening and the sintering of these particles as a function of the cycling times. And then afterwards, they take those chips out and then they can look at them using, again, high resolution without the liquid in the way to look at, the atomic resolution changes that they're seeing. And so in general, you're always going to be combining, both pre and post ex situ results, other correlative techniques with your in situ results in order to really effectively interpret them. So one of the biggest obstacles that people first encounter when they're working with liquid TEM is the effect of the beam. Now, certainly, no one who works with an electron microscope is a stranger to beam interactions. Knock-on displacement, diffusion of atoms, radiolysis, And electrostatics and heating are something that samples are always subjected to. That's one of the reasons that cryo is so widely used, is that the low temperature helps to mitigate some of that, particularly in very fragile systems. But when it comes to liquid, radiolysis is incredibly important. And that's because we're no longer in a vacuum. So not only do we have radiolysis and damage to our sample from the beam, but we have decomposition of our liquid that we're using by the beam, and that can create a lot of reactive radical species. And so the example that I really like is, for instance, water, which is very simple. It's used, probably in about, 85% of, the published results is, aqueous, aqueous solutions. But with when the beam interacts with that, it creates this cascade of highly reactive species that, again, can now start to interact with our sample and drive chemical reactions, cause degradation, cause new side reactions. And so Francis Ross did a lot of really wonderful seminal studies of what the dose is doing to our solutions. And generally, most researchers who are doing this type of modeling stick pretty much with the with looking at water, because other systems are really complex, or they look at the radiation chemistry literature to see what types of radiolytic products they should expect from their solvent. And so what she really showed is that as you increase the dose rate, you're going to increase the concentration of these reactive radical species. But if you hold the dose rate constant, you'll get a steady state of these. And you can model this. There's a lot of really interesting studies that have been done where people have, there's a lot of models out there. There's a lot of scripting out there. People are really, they're really delving into this. But we have to think about this when we design our experiments. And so this is a really nice figure that talks about the types of things you might see and the dose rates. And so if we look up here, we've got high resolution way up here. And so you will very rarely see true high resolution in a liquid cell that is really full of liquid. And by full of liquid, I mean, 100 nanometers or more. And so the acceptable dose to the sample is, really the dose at which, that you can still get the resolution you need without damaging it or causing things you're trying to, avoid happening. So bubbles are one of the most common things that people see, is particularly when they try to go to these higher dose rates to get high resolution, is in water, you're going to see these hydrogen bubbles forming. And very visible, they form very quickly. If you turn the beam off, generally they will dissolve back into the solution. And so that's something that you have to look out for. This is a solution of gold nanorods, and you can see in this case, the beam was intentionally condensed in order to generate a high dose rate and generate these bubbles. And then the other thing that's really important is that at doses below bubble formation, you can get metal ion reduction. So if you have a solution that has metal ions in it, you can reduce those and you can essentially grow nanoparticles. And so in this case, this is you can tell by the square here that this was done in scanning TEM mode, so in STEM. This is, I believe it's just a solution of gold chloride and water. And what they've done here is they've imaged for a while at a dose rate, sufficient to reduce these ions. And they've preferentially essentially grown on the surface of the silicon nitride window. When they zoom out to a lower dose, that slows the growth down because, of course, the dose is much lower. But you can see the square really nicely where the beam was and where that higher dose was. And so, that's something you have to really think about when you're developing these experiments. And so there's really, two strategies and that people, take depending on what they're trying to do. So either you minimize the dose to where it is not affecting, the part of the experiment that you're interested in. And so an example here would be, this is some work from Leila Mehdi and Nigel Browning's lab looking at the decomposition of different battery electrolytes and at what dose they start to see decomposition, see particle formation, things like that. So they calibrate their electrolyte or the solution that they're going to work in by exposing it to a dose range and determining sort of what's the minimum acceptable dose. And then they have to work in their experiment and stay under that dose level. The other strategy is to exploit the beam and use those reactive species to drive reactions. And we see this a lot in studies looking at nanomaterial growth, because essentially the beam is acting as our reducing agent. So you'll see some examples of experiments where people start with different template particles in a solution of metal ions. They're using the beam to essentially reduce those ions onto those templates and see what's happening. So this is kind of a really classic experiment. It's one that you see a lot in demonstrations. It's one that if you're just starting out, people, when they're just learning, they'll usually do an experiment like this. You can use many different solutions. In this case, this is a solution of gold chloride. And at low electron doses, you get more reaction-limited growth. And so you'll typically see that you have more control over the shape, the size of the particles. Whereas if you then crank the dose up, you're going to get diffusion-limited dose, because you're going to be reducing all of those ions immediately in the vicinity. And it takes a while for new ions to diffuse into that liquid. And you see a much more dendritic or sort of popcorn-like structure. So that's sort of what you have to think about. You always have to have dose on the mind. So you want to really minimize your dose between imaging. Don't leave it, the beam on while you're setting up file structures and things to save. Always, there's a lot of strategy of blanking your beam, taking your measurement, keeping the dose rate low, moving to different areas to ensure you're looking at pristine regions. And but now we can kind of move on. We can talk about, we have enough background, we can talk about the experiments that people are doing. So this is a really nice example looking at using the beam to study how structures form, what will how changing different reagents can affect our end product. So there's a lot of studies looking at structure function relationships and what are the mechanisms that control those. And so in these examples, this is a shape evolution of different silver nanoplates. So starting with different shapes in the presence of a capping agent, which is dimethylamine, and a coordinating complex, potassium iodide. And these are different experiments where using the beam to produce the solvated electrons that will then plate these gold ions onto the silver nanoplate. And they picked the concentrations in order essentially to slow down the growth, because it has to be slow enough the camera can capture it. And so what they found by looking at these different systems, different concentrations, and they looked at a wide range of concentrations for each of these, is that the dimethylamine was really responsible for tuning that growth rate, whereas the potassium iodide had a much stronger impact on the final morphology. And so they were able to tune the compositions of that in order to improve stability of the particles. And what they're really trying to figure out is what is controlling these mechanisms during the growth phase. And so they found that the nanoparticle shape very strongly dependent on the monomer ratios, deposition versus surface diffusion. And so there's a lot of different examples like this in the literature. Another similar type of study, in this case, they're looking at the effect of the surfactant concentration in solution. And so in this case, these are, I believe they are gold nanoparticles as well. Again, from a solution of just gold ion. But in this case, has different concentrations of CTAB. So a solution that is just the gold chloride in water, the 7.5 millimolar CTAB and 50 millimolar CTAB, the same dose rate, the same time scale for each one. And so you can really see very clearly here the effect the surfactant has on controlling the size distribution and growth rate. And so they were able to, measure that and determine, some of the kinetics. And so something like this could be used to, optimize an experiment and understand better what's happening. moving to a more complex type of system, but still, looking at, growth. In this case, this is biomineralization, looking at things like hydroxyapatite. In this case, they have a solution of essentially hydroxyapatite precursor in artificial saliva. And they're able to see within the liquid cell that you have these ion poor and ion rich phases. And over time, again, the beam, of course, is having some impact, in terms of speeding up the reaction, acting as a reducing agent. And they're able to observe the formation of the hydroxyapatite nanoparticles. And what they see is that, both classical and non-classical growth nucleation pathways coexist in the system. And so these are some movies taken at different points, different regions of the cell. And so what they see here in the first one is you have in a supersaturated solution in those liquid separated regions. This is they're seeing the direct nucleation and aggregation and coalescence of the hydroxyapatite. And the movie is a little bit long, so I'll let it play out. But in the same experiment, in the same cell, they're also seeing examples of non-classical nucleation and growth. And so in these examples, you have the hydroxyapatite is nucleated onto ACP substrates, and then it's growing and coalescing, and you have dissolution and detachment occurring. And so both of these are happening in the same system. And so from that, they were able to essentially propose, models of what was happening, and that you really had this very complex growth pathway occurring in some of these, biomineral environments. Another example here, this is a soft material, is looking at growth of polymeric nanoparticles. So in this case, they're using, it's a much more complex system than just metallic ions, and so they're looking at it's a polymer, a block copolymer system. They have I believe it's a DMA, and so they're watching the formation of these monomers into these larger particles, but they're also using variable temperature. So they need to have the solution at above 70 degrees in order to see the assembly of these polymeric amphiphils that create these warm like vesicles and then ultimately form into micelles. And so, of course, with these very low contrast soft materials, it's quite difficult in liquid with the liquid thickness to really get significant contrast. So they were able to visualize and watch the self-assembly. It doesn't start to occur until it reaches the temperature of 70 degrees, but then they start to see the formation of these particles. And they start to see the spherical particles forming, and then the development of these sort of worm-like micelle structures. But that's not necessarily for some of these types of experiments. In situ liquid is not going to give you the full answer. But what they did, which was really interesting in this study, is they took the cell apart afterwards, and then they did MALDI mass spec on those chips, and they mapped that whole chip to see what the molecular weight of and characterize the polymeration that occurred post in situ experiment. And so that was a really interesting example. And so far all we've looked at are essentially just growth, in this case growth with different temperature, but one of the areas that we see a lot of interest in is electrochemistry. Electrochemistry is really very important in situ liquid experiments. And so one of the challenges, of course, of that is it's fairly straightforward to put electrodes onto the chips and deliver electrical signals to the liquid, but you want to really be able to measure that and compare that effectively with the results of benchtop studies. And so that means that we have to do a lot in order to make sure that we know that we're putting in clean current. We're not having a lot of crosstalk, and we need a really good way to reference that. So the reference electrode, of course, is very fundamental to any electrochemistry experiment. But we are limited for in situ in terms of the types of electrodes that we can physically put onto the chip, because it has to be compatible with semiconductor fabrication techniques. And so most designs utilize a so-called or quasi reference. And so the quasi reference, in this case, platinum, they're commonly, they are used in electrochemistry, particularly common with non-aqueous samples, but you need a way to kind of compare that to the literature data. So either by using internal calibration or calibrating those electrodes in order to compare them to other benchtop studies. So there are a couple of ways that you can do this. So you can do, for example, an ex situ calibration where you calibrate the reaction on the benchtop with respect to your normal electrode. In this case, this is an example of using a reversible hydrogen electrode, and then calibrating it with a platinum wire and determining what that shift is. And then you can take your in situ data and adjust those, shift the peak values accordingly, and see how to compare that with literature values and benchtop experiments that use a conventional electrochemistry setup. Another method is to use an sort of internal calibration, and this has the advantage that you can do it in the in situ cell. So in this case, they needed to determine the potential shift between their platinum reference electrode of the E-chip and a lithium metal electrode that they're using in their actual setup. And so what they did in this case is they used just ferrocene, which is very standard. They look at the redox couple peaks for ferrocene when they cycle the experiment using the lithium electrolyte that they're going to utilize in the experiment, and they take the half-wave potential of that to determine what the shift is, and then they can shift all of the peaks that they see in their in situ experiment with respect to the platinum back to the lithium electrode. This is, again, extra steps. It's kind of cumbersome. You have to, make sure everything is calibrated correctly. The other option, and we worked, very closely with Rob Kelly at University of Virginia, who does a lot of corrosion studies, to integrate a different method of using standard reference electrodes for in situ. So you can use what's known as a metal bridge. So most benchtop type studies will use a salt bridge, and the salt bridge allows ions to pass between your cell that has the working electrode and counter electrode and the reference solution. They're all generally in something here, together, but there's a salt bridge in here. In this case, we're using a metal bridge. Now the metal bridge here is platinum. That's the same electrode that's on our chip. So rather than having to calibrate it, we simply use the platinum reference electrode on the chip as part of the metal bridge into an external reference electrode. And you can use any off-the-shelf reference electrode. So that really makes it much easier. This is some results showing that, the metal bridge is, works, it gives you the same results as the conventional setup on the bench top, and that the length of this metal bridge, because remember, the holder is in the microscope, the metal bridge has to go all the way out of the microscope, is not affecting the electrochemical results. And so this is an example of without the metal bridge, and with the metal bridge, everything shifts over, in this case, to where it needs to be with reference to a silver chloride electrode. And so all of that external electrode is housed in a Faraday cage that sits very close to the goniometer of the microscope. And so this way, you're able to really get very, accurate electrochemistry results. And of course, within the literature, you're going to see all of these different methods used. So this is an early example looking at battery charge and discharge. And so this is, in this case, this is a platinum electrode. They're cycling the potential first in the negative direction. There's a lithium electrolyte in solution. And as that potential shifts further and further to the negative, we're going to see the plating or as it discharges onto the electrode, and then as it cycles back, some of that redissolves into solution. You'll notice not all of it redissolves. And so if we do a second cycle here, the same thing is going to occur, but we already have some lithium now left over on the electrode. So more lithium is going to plate onto that. And when it cycles back then more and more is left. So this is a really, very good visual example of capacity fading and the degradation of battery lifetime as you do more and more charge cycles. Another example, this is the same group, same system, but in this case, what they're really looking at is, well, how do we optimize the electrolyte so that we don't have as much dead lithium left, so that these the charge discharge cycles are more we're not losing a lithium here, so we have better efficiency. And so they looked at water as an additive, and what they found is that with the 10 parts per million versus the 50 parts per million, more water in solution, while it resulted in larger grain size, resulted in much better dissolution and increased efficiency. And so they characterized this from the movies that they obtained in situ. So with the water additive, they had a faster and larger deposition, but a more less residual material left after dissolution. And so they were able to attribute this to the the formation of the lithium fluoride that occurs when the water is added to this electrolyte. And I'm going to skip ahead a little bit to a recently published study. So this just came out this fall, and it's a really nice example of using in situ electrochemistry to really correlate specific structural changes on the surface of catalyst fuel cell nanoparticles to the peaks that are seen in the cyclic voltammetry. And so this is a core shell octahedral nanoparticle. So it's a palladium core with a platinum shell. Core shell nanoparticles are very, highly widely used because you use much less platinum if it's just on the shell. So it is much more cost effective. You can really tune the shape and the size, for different reactions. And so during the active catalysis, you have the molecules are coming onto the surface of the catalyst. They're creating short-lived transient states. Bonds are breaking and reforming, and over time, the catalyst surface can degrade, which is going to impact its activity. So understanding what these mechanisms are is very, very critical. And so this is some ex situ results, kind of with the pristine particle. And then after a durability test, they did 50,000 of these high potential cycles. And you can see significant structural changes, and also they're losing a lot of activity. So what they want to find out using in situ is what's happening between these two states. What is really going on? And so to do this experiment, they did in situ electrochemistry. So first, they did a beam control with no cycling, no electrochemical cycling, to determine what the optimum optimal beam dosage was, to so that they weren't seeing degradation of the particle from the beam. Then they looked at a wide range of different potentials. So in this case, this is a cycling over negative 0.5 volts to 0.5 volts. And what they realized as they were doing these studies is that if they shifted to lower potentials, they were really able to slow down the corrosion rates, which meant that they could capture it more efficiently. In situ, it wasn't happening so fast that the camera can't keep up. So this is ultimately, they used a so when they moved to about negative 0.2 volts to 0.2 volts CB, they saw the formation of these sort of islands and particles on the surface of the catalyst particles. And so what those are is that these slightly the initial CV of 0.2 to minus 0.2 to 0.2 volts, that drives the formation of these small platinum islands on the catalyst. So it sort of creates some imperfections. And since they're really, what they're interested in is studying the corrosion of the palladium core, they applied this to, sort of quickly drive some imperfections. And then they switched to a lower potential range, in this case, minus 0.9 to minus 0.2, to really slow down the corrosion so that when they did a full cycle, they could capture the entire series. And so what they did then is they, did their CV, and they were able to correlate the different structural changes to different points in the CV. And these islands here are those sort of imperfections on the surface that they were seeing. And so doing some, post-analysis afterwards, they were able to identify, the initial islands are platinum that's redeposited, and then it starts to build up with palladium that comes out of the core. And so this first peak here, peak 1, this is the reduction and deposition of palladium that gets exposed to the electrolyte on the surface of the platinum shell, and particularly at the edges. And then the second peak here, this is really dominated by, once those that shell is damaged, then the palladium really begins to corrode. So this peak here is the palladium etching, and then peak 3 here is more etching of the palladium from deeper within that core, as well as some additional platinum surface corrosion. So the platinum corrosion is more likely to occur at higher potentials, so during like startup or shutdown of fuel cells, whereas the palladium core is going to be more likely to be corroded during the actual running of the system. And so using that, they were able to really applying more cycles over time and looking at how these peaks shifted, they're able to build a better understanding of what is happening during those multiple cycles to get to that final product. And so again, here's this is our peak 1 down here. And so as the it's pretty stable, it stays about the same point. So this is our reduction and deposition of palladium on, particularly on the edges and the corners. What they find is that those are more stable regions, so they don't tend to etch away completely. Peak 2 again here, it is experiencing a negative potential shift because as it cycles more and more, the palladium becomes weaker and weaker. And so more and more, the platinum encapsulation becomes weaker and weaker, and more palladium under the shell is exposed. Peak 3 here, we're seeing a positive potential shift. And so it's getting harder and harder to oxidize those platinum molecules on the shell because you're forming platinum oxide over time. And then this one here, this peak 4, this is actually the first cycle, and it's the tail end where they did that initial higher potential cycle to damage the platinum shell essentially. So this is the residual of that. But again, the platinum shell, once you kind of do that, at that point, the palladium corrosion becomes the defining factor. So what they also found here, looking at this, looking at the shift between here, peak 2 and peak 3, this is the the potential difference, between the surface and core palladium etching. And from that, and the ex situ studies, they were really able to identify the different stages that occur with these octahedral core shell nanoparticles during the cycling, and for the first time really correlate the specific high resolution surface changes to the what's happening in the electrochemical spectra. And so they the corrosion process, once you have some of that shell damage to where there's some palladium exposed, you begin etching the inner palladium, and then both the palladium and the platinum will redeposit onto the exterior surface during that reduction process, and it will redeposit preferentially on the corners, and so those will stay, build up more palladium and platinum, and then the terraces, so that's why you have these strong, very thick layers at the edge, the corners of these octahedral particles. So with that, I know I'm running a little bit out of time. I'm going to show you just very briefly 2 results from our new system. So we've been spent the last year and a half or so, we've really worked on widening the temperature range. So the previous results I showed you, that holder can only work between room temperature and 100 degrees Celsius. What we have developed now is a new holder that can apply temperatures from -50, so you can look at freezing studies, you can look at low temperature organic solutions, up to 300 degrees, while doing simultaneous electrochemistry. So before, it was either you could do heating or you could do electrochemistry, but you can't do them both at the same time. And so this was a lot of optimization, both for the safety to make sure that the holder, was able to, with these temperatures, not melt, and and particularly eliminating the crosstalk, which is a very important thing. So when you have, I'm just going to skip this one in terms of loading the holder, so this was an initial a prototype system, and what happens here is this is what happens when you have a lot of crosstalk present. As you go to higher temperatures, which require a lot of power, you see really big shifts in the open circuit potential, which is going to completely mess up any type of electrochemistry measurements you're trying to make. And so we went back to the drawing board and did a whole looking at the holder design, the connections and contacts, the chip design, probe skating, goniometer bias. And so this is showing with the minimized crosstalk, you have these very, very small changes in potential with fairly large changes in temperature. And so this, we are confident now that we can get real measurements even at higher temperatures. And so this is some early examples. This is some work done with Yao Yang at Cornell. And so in this case, this is at 98 degrees. We're looking at I believe it's a copper sulfate solution. And so we can see a lot of plating here on the surface. And if I play the CV here, we have this really strong plating and dissolution. Now, if we do the same experiment and we take the temperature down to, in this case, 5 degrees Celsius, very, very little plating. I mean, there's maybe something happening, but very very low. The other thing to notice here is that we have a much higher dose rate. So at these lower temperatures, we are able to use a much higher dose. Now, is something happening? So if I play this here, it looks like really nothing is, but that's just because we have the same scale set here to at 98 degrees. If we zoom in, we actually are seeing plating at these low temperatures. It's just very small. So, this is important for, a lot of, battery and fuel cell development, because again, these devices can fail at both high and low temperatures, and understanding and optimizing their performance under these conditions is very important. And then the other one that I wanted to show you just very briefly, I'm going to see if I can get the video to play. This video does not like to play very well. So let me see. So this is a freezing video, and unfortunately, I don't think it wants to play. It's not going to go. Let's see. All right, well, I'm just going to do this part here. But what we're seeing as we take the temperature down, and for whatever reason, it is not syncing up, and it always works perfectly when I test it, and then as soon as I put it like in presenter mood, it doesn't want to do its thing. But what we're seeing here is the freezing, and we can see the grains forming. You'll also see these interesting bubbles here, and these bubbles increase in size because as it's freezing, it's pushing the air out of the water here. And we see that changing the focus here a little bit. We can see the faceting of those bubbles. And then those bubbles continue to grow as it freezes. And then if we go back up in temperature, let me see, they'll start to melt back and then goes back to solution. So that's just a little bit about where we're kind of the next steps that we're taking with some of these in situ liquid systems. And I hope that this has been an interesting look at something that you can do to increase what types of studies you can do with a TEM.
Thank you very much, Madeline, for this very interesting talk. And it is amazing to discover all this application that you can do with this liquid holder for TEM. And yes, we can start with a question. Is there already some question? Yes, Shaksi Babulai Schmidt.
Yes, hi.
Please.
Can you hear me?
Yes.
Hi, okay,
yes.
So really nice presentation. And my field of research is actually biomineralization. So I was very interested in that paper you showed from Science Advances about biomineralization. But since then, a lot of good studies have come out with aggregation of hydroxyapatite and calcium phosphate. But I'm wondering, where are we in terms of introducing a biological component like a protein, collagen protein, in actual biological simulation fluid? Where are we in terms of that?
People are working on that. I don't know that we have any published results. I do know several researchers who are, actively pursuing that. So it's definitely something that people are really looking at. There's a nice paper that will come out hopefully soon from Catherine Granfield and Liza DiCecco and McMaster. They're looking at biomineralization on like bone implants. So they actually have a fib sample in the holder and they're flowing, they've got, stuff in there to look at that. I believe I think they have a communication out, but there's a full paper coming out. So there has been there were some early studies looking at, say, seashell formation type protein from, I'm trying to remember the name of it, but I can't offhand, from Roland Kroger, looking at you added protein to the solution, how the calcium carbonate morphology changed. But I don't off the top of my head know of any published results yet with the collagen, but I can think of at least like 3 groups that are actively working on those types of studies.
Okay, good to know. Thank you.
Yeah, thank you. Is there any other question from the audience? I have a question for you, Madeline, concerning biological sample. That is because yes, using the dropcast method, we are able to create different thickness of the liquid in the micro wheels in the bottom chip and then after we cover with the top chip. But the question is, when we work with flow mode, do you have a method to control the thickness, or because the thickness is very important for biological sample, and then the signal is really weak.
Yeah, so one of the things that I know you could try is to use a different type of pump. I know that some of the pressure type pumps, which work just fine with our system as well, will allow you to kind of push and pull a little bit. There's a paper from Stefan Merkens, and I'd be happy to send that to you, that looks a little bit at that, trying to sort of mitigate the pressure for flow. So that would be one way that you could start there. And, of course, window size. But yeah, so the flow with some of the, ultra thin layers is difficult to control a little bit more because, again, you've got liquid flowing in and it's going to press those windows apart. So certainly the smaller the windows are, the less of that bulging that you're going to have. So that, but I would probably maybe look at some of the trying to apply some differential pressure to the cell to kind of keep, you know, the liquid, you know, from pooling as much.
Okay, thank you very much. Any other question from the audience? I'm checking the list. Okay. Yes, no. Well, if there is not a last question, maybe we can close here. Thank you again, Madeline, to accept the invitation, and thanks to the audience for the participation. Thank you.
Thank you so much. I appreciate it. Bye.
Bye.